As a reminder, collect 25 microliter samples at indicated steps of the purification to analyze by SDS page. To begin with, obtain your cell pellet. Transfer your thawed cell pellet to a beaker containing a stir bar. Measure out 5 to 10 times your cell weight in buffer A, which is your lysis buffer, and add it to your cell pellet. Place the solution containing your buffer and cell pellet on a stir plate and stir the solution until it becomes homogeneous. Place your cells on ice. Set your sonicator up so that the horn or the tip of the sonicator is about 1 centimeter below the top of the solution of your cell solution. Ensure that your cells remain on ice throughout the procedure. When sonication is complete, remove the horn from your cells. Wash the tip of the horn with ethanol and wipe it down with a Chem wipe or a paper towel. At your bench, pour your lysate into a high-speed centrifuge tube. Balance your tube with another high-speed centrifuge tube filled with water. Place your balanced tubes on opposite sides of the rotor in a centrifuge. Apply the lid to the rotor, making sure that it is tight but not too tight. Close the door and run a program provided to you by your TA. When complete, open the door, remove the lid, and remove your tubes. Note the cell debris or the insoluble portion of the cells are at the bottom of the tube. Pour off the supernatant into a clean beaker. At the FPLC, begin operations by turning on the instrument. The Unicorn software will be up and running and will automatically connect to the instrument. Begin by double checking that the lysis buffer is in buffer A and that the elution buffer is in buffer B. Next, we will attach our 1 mil His column, or any column for that matter, to the FPLC. First, we unscrew the coupler to the column platform and screw it to the top of the column. Next, we screw the bottom of the column to the top of the platform. In Unicorn, if the instrument is properly connected, you will see the green lines lit up. If you are the first user to the instrument, be sure to pump wash both buffer A and buffer B. Begin the manual program by beginning the flow rate at 1 mil per minute. Add a pressure alarm so that the instrument does not become overpressured. Be sure the instrument is running by monitoring the UV trace and the conductance trace. As the column is equilibrating, load your sample into the super loop. By injecting it with a syringe, load the sample into the syringe and be sure not to have any air bubbles in the syringe. Connect the syringe to the injection port and carefully inject the contents of the syringe into the superloop. Notice that the plunger in the superloop will begin to rise. Change the injection valve to inject. Changing the injection valve to inject changes the flow of the system and pushes the sample through the column, through the detector, and into the fraction collector and out into the sink. Do not allow the plungers in the superloop to meet. This may cause damage to the superloop. Before the plungers meet, change the valve back to load. Changing the valves back to load allows the lysis buffer to flow through the column once again. This will clean out any unwanted, non-specifically bound proteins to the His column or to any other column. Wash the column with a minimum of 5 column volumes. Dilute your protein, change the concentration of buffer B to 100%. Fraction collector. Set the fraction collector up so that it drops into the first test tube. The fraction collector will automatically collect in volumes that you indicate on the FPLC. Start the fraction collector with the appropriate volume of fractions. When you are done with the fraction collection, hit End in Unicorn and that will shut off the FPLC. Remove your fractions from the FPLC and take them back to your bench. Pipette your fractions into clean microcentrifuge tubes. Label the tubes and place them in the freezer.